All right. Well, hello. I'm Sarah Talarvik. I'm a biology staff scientist here at Vernier, and I'm a former college professor. I taught for about 15 years.
Hi, I'm Colleen McDaniel. I am a biology and environmental science educational technology specialist, and I've been at Vernier now for 7 years. And I used to teach in Texas, where I taught pre-AP biology and AP environmental science.
Hi, my name is Damon Tai. I work for BioRads Laboratories as a curriculum and training specialist. I've been there almost 10 years now, and prior to that, I worked at the National Labs on the Human Genome Project, and prior to that, I used to be a high school teacher in Portland, Oregon.
All right. Well, I'm going to ask Damon to tell us a little bit more about this BioRad kit that we're doing today. We're going to be using the Algae Beads exercise for photosynthesis. And Damon, go ahead and take it away.
All right, I'm going to share my screen with you guys. I'm going to take you through a couple slides about this kit, kind of why BioRad decided to make this, and then some of the functionality behind it, and a little bit of like the tips and tricks too that I've learned after using this for a number of years. So I'm going to go ahead and share the screen. So BioRad built this kit, boy, I think it was, and this took quite a few years to get out the door. I think it actually launched about 3 years ago. It was mainly because we were looking at AP biology test scores, which should give you a pulse on how the brightest biology students at a high school are doing with concepts. We were seeing that about half of the students in AP biology were coming out thinking plants didn't do cellular respiration. And being a former high school teacher, I was like, oh, I know we're getting that in the theory portion of the class. Why isn't that sticking with them? And it's probably because traditionally we've always done 2 separate labs, one for photosynthesis and then one to model cellular respiration. And so our goal was to basically get those into the same lab. So that way it got rid of that kind of confusion that students were walking out with. And the beauty from a teaching point of view is you get to consolidate 2 labs down to 1, which also saves you time and hopefully just energy as well. So the solution we came up with was using these algae beads. So we've taken a freshwater algae and basically polymerized them into these small, easy to transport beads. These are just the right size so you can transfer them with one of those plastic transfer pipettes if you kind of modify the pipette by cutting it right in the middle of the barrel tube. The beauty about them is they can be reused multiple times. That's what I really like about this lab, and is that you can take that same kit and use it for multiple classrooms because really nothing's being created or destroyed during each of the events that students use them. They've got a shelf life of about a month or more after you've activated them. By activating that just means bringing them into the light for the first time. And to be honest, like that's what BioRad kind of guarantees is that month. But I've been able to take sets and get them to go for, I've got one set right now that's almost at 8 months in my bathroom. They have a 3-month shelf life from the data delivery. And then the algae beads are safe to dispose, which is nice because sometimes some of the stuff that people were doing photosynthesis with, you had problems with disposal depending upon where you were at in the country. So to make it safe to dispose, we specifically went after an algae that you basically see everywhere, at least in North America, which is Senodesmus obliquus. So just to get a pulse on that, I go to my favorite website, inaturalist.org, which is a great way to see species biodistribution. And this data is all generated by just users like me and you using their cell phone. And so we can see here that Senodesmus occurs all over kind of North America. So there's really no constraints when it comes to kind of disposal. The only asterisk on that is if you're in Hawaii, Hawaii is really good about trying to protect their environment. And so they have a little bit of extra paperwork you've got to jump through just to import this algae, even though that it's already there. The other side of this lab that makes it work so well within a classroom is you've got those algae beads, and then you've got this really tightly built, basically pH indicator that tells you how much kind of carbon dioxide is coming in and out of the solution. So this indicator allows you to monitor when the algae is doing respiration. So if it's in the dark, that's the only game running and it's dumping CO2 out into the solution around it. And it'll take this pH system that's usually kind of this barely kind of ruddy, reddy color around like, atmospheric CO2, and it'll shift it to be yellow in a number of minutes. And then if you were to take those same beads in the solution and go into the light, they're still doing cellular respiration in the background, but they're really driving hard on the photosynthesis. So they're gobbling up all the carbon that's in the water and then also dumping O2 out. And so you'll actually get a pH shift upwards, and this will turn a deep purple color. So it's actually really easy to monitor what's going on and it happens really fast. So usually within most classrooms, I see that they can kind of do this within 15 to 20 minutes. You'll get that kind of full movement to both sides of the spectrum, which makes this really easy to pull off. And then just from a prep point of view, for any of you guys that have worked with BioRad stuff, a lot of our stuff, it takes a decent amount of prep because you're doing some relatively complicated stuff in your classroom. This by far has to be the easiest BioRad lab to prep. I actually love it when I have to do this one in person because I know my prep time is just going to be 15 minutes maybe. The kit itself that BioRad sells has a whole series of different experiments they can do using these beads. So they can actually take the beads and actually get rid of the polymer in there so that they can look at the algae underneath the microscope. And then you can kind of do what we're going to look at today, which is just kind of comparing a light and dark reaction to see the rate at which the pH changes. And then building upon that, scaffolding from there, students can do a whole host of different experiments, figuring out how the intensity of light changes those rates. How does, say, the spectrum of light change those rates, and temperature around them? Because those are big questions if you think about this, especially within kind of a climate change context. Because imagine what would happen if, say, changes in temperature shifted, say, the rate of respiration faster than the rate of photosynthesis. We would all be in a giant world of trouble. So students can compare how those rates shift. Do they shift together under all these different variables? The kit itself is $145, and it's enough. BioRad always designs around 8 lab stations, so it's enough you can set up 8 lab stations and have students run those. But they actually give you so many beads in this that I find whenever I get this kit, I'm usually able to set up between 10 and 12 lab stations. So know that that's part of the build in there to make this for sure workable for your classroom. It also comes with these color indicator cards so that you can use that as just visual checks to see how things are moving. But man, if you got the spectrophotometer from Vernier that you can use to do the mathematical analysis with this, that's really what you want to be doing, because those are the skills, trust me, coming from industry, that so many of the employees that I was hiring were missing. You'd ask them to set up like a standard curve or do some sort of analysis with any sort of standard curve, and it was like hand-holding for a lot of these 4-year grads. So this is something you want to make sure they can kind of do, and that's why using a spectrophotometer is big. BioRad a lot of times puts things out there without a great context for them, and so this one actually, we've got a couple slides that you can pull off our website that have ways to put this within a context to make students care about why they should understand these different rates. And so one of them is kind of a phenomenon-based thing, where I'll basically show students this picture, and we'll just kind of build from here and ask them, like, what's going on? What do you see here first? And then what do you think is going on? We come up with some hypotheses, and then I'll add more information kind of to their brains and say, okay, let's back up. Where this is going on is down here in Atchafalaya Bay in Louisiana. And you can see from this, we've got some new information. We can see, here's the Mississippi coming down. We can see kind of like a green hue in the water. What is that? And some kid will eventually go, oh, maybe it's an algal bloom. Like, all right, yeah, it is. Why would that be bad though for the fish? Because I mean, algae, right? It's growing, it's taking down sunlight. Shouldn't that be great? It's more food for everybody. And you can kind of dig then into how some of these algal blooms at night kind of flip over to be dangerous situations where you can have basically O2 crashes in the water because all of those photosynthetic organisms all of a sudden at nighttime are gobbling up all of that O2 that's in the water, and you get these kind of like hypoxic zones on top of some of that decomposition that's going on as well. What I would really encourage people to do with this though is find a local example of these algal blooms that are causing problems in your area and use that as the context to then pull students in. So if you're in the Pacific Northwest, I mean, Hood Canal, that's the one to go to. If you're in California, you got the Salton Sea. There's usually an example, unfortunately, just about every place in the country. If you need help with that, I'm more than willing to help you find one in your area. You guys can just send me an email and I can help out with that. The next few slides are just some kind of tips and tricks I've learned from using this over the years that BioRad doesn't necessarily have in their documentation, but I just kind of want to get out there. So as you're playing with this kit, these are things you might think about doing to make your life a little bit easier. So one of the first things is prepping the indicator for this. BioRad has you make this 1x pH indicator based upon a 10x indicator that they send you. And it's basically mix it up and then let it sit for 3 days. I'm a really impatient person, which BioRad doesn't always like. So I'm like, 3 days, oh my God, I can't take that. How can I make this faster? This is a surface area problem. So I'll do is I'll mix up the 1x indicator and then just pour it back and forth between cups a whole bunch of times until I see it stop changing. And then I'm pretty much ready to roll, and I saved myself a couple days. The other thing is for kind of transferring beads, what I found to be the easiest thing in activating the beads is I'll put them in a Petri dish with some of the pH indicator after washing the beads with water just to remove the storage solution they're in. And then I can just expose that entire Petri dish to light, which allows me to activate them really easily. And then I'll just I'll cut a transfer pipette right in the center, so that way I can easily move beads and kind of count them to move them into containers for the students to play with. The other thing is BioRad provides you with this box full of cuvettes, and you use about half of the cuvettes potentially for this lab. And so what I like to do is I like to take that box and then make that as a self-service rack where students now can grab everything they need, and I don't need to put out anything. So I just pull out the box every class period and they just grab from there. So I'll basically make 2 cuvettes with beads in it, one with pH indicator, one with water so they can wash the beads, and then a cuvette for waste. They grab those, they go to their desk, they can run all the experiments, and when they're done, they just re-rack it, and all I have to do is resupply the water and the pH, and we're ready to go for the next class period, which is really nice, makes it really easy to perform. The other thing is those color cards you get, if you're going to use those, it's kind of just a way for them to kind of background scale where things are moving. Put them in Ziploc bags. You'll never have to buy the whole kit again. And you can literally just buy the refill kit every year from BioRad to make it really easy. I also like to make just pH buffer visuals for the students, just so that they, instead of just looking at that cue card, it's nice to actually see it in the cuvette. So I'll take that indicator, add a little bit of vinegar to one of them, squirt a little bit of, like Windex in the other one, and that way it'll drive it to both ends of the scale, just so that way they've got nice visuals of what their cuvettes could potentially look like at the end of the processes. Another kind of thing that you might want to think about is that BioRads Protocol has you put 10 beads in the cuvettes. And with 10 beads, you end up with a 3x3 grid of beads on the bottom with 1 bead that hangs out on the top. So sometimes if I know my spectrophotometer is a little finicky as far as where the beam path comes through, I'll just have students do 9 beads instead of 10. It makes the reaction just a little bit slower, but then I don't have to worry about that, my light beam hitting that bead. And that's basically it as far as kind of the tips, tricks, and the backgrounds on this lab. So let me go ahead and stop the share of my screen, and we'll turn it back over to Sarah, who's going to take you through how to actually run this on SpectroVis.
All right. Thank you, Damon. That was incredibly helpful. So I'm going to start with, I've already done some of my prep ahead of time, which is labeling my cuvette tops, as well as getting my pipettes ready for transferring algae beads. So at this point, I'm going to switch to using the handout that we've made available, where you use the rest of the BioRad kit, but you get to the part where you want to analyze using Spectrophotometer. And one of the advantages to using our spec, I'm also wearing my PPE, since we do recommend wearing goggles for this lab. The GoDirect SpectroVis Plus can be used for analyzing your samples. So I've got the BioRad card here as a reference, and then I've got my cuvette set up in a cuvette holder, labeled and ready to go. The first thing we need to do is transfer some algae beads into the first cuvette, which we've labeled A. So we're going to count out 10 of those. And these algae beads were prepared by letting them sit under a, we're going to actually use an LED grow light today. All right, a few more there. I believe that's 10. All right, we do want to remove any excess liquid. So we're just going to remove that with a pipette and put it in a waste container. And then we're going to transfer 2 mils of the indicator. We want to try to do it without getting any bubbles in it. I normally wouldn't be doing this over the spec. I wanted to make sure we got it in the field of view of our camera. All right. So we're going to just prep that and get it ready. What we want to do next is we want to launch Spectral Analysis. So we'll be using our free app for spectrometers from Vernier. I'll get this launched. And then I'm going to share my screen so that you can see it. All right, so I'm showing my spectral analysis screen. And for this lab, we're going to pick absorbance versus concentration. Even though that seems a little bit counterintuitive, that's going to allow us to do a data collection mode that is essentially sort of an events with entry mode. So we're going to select that mode, and then we're going to be prompted to wait for the spectrometer to warm up, and we'll calibrate. So we're going to give it the warm-up time. Notice that it has a countdown available, so we know exactly how long it will take. And then I have a blank cuvette prepared. We'll give it just a few more seconds here. And in the meantime, we can gently wipe the sides of our cuvette with some lint-free tissue. And then we'll place the cuvette in the device. Finish our calibration. All right. And now we can prepare our test cuvette with our liquid. So rather than put the entire cuvette in the spectrometer, I usually transfer the liquid out into a test cuvette. So we do the incubation with the algae beads in one cuvette, and we do the testing in another cuvette. And this just ensures that there's nothing in the line of the light for the spectrometer. We don't want anything in the pathway of the beam. All right, so we've wiped the sides of this one. Make sure that there's no bubbles. And then we'll place our indicator in the spectrometer. The wavelength that you're looking for is 550. So we're going to input that. All right, and this is actually where we're going to take our very first point. So we're going to start collection. We're going to make sure that our signal is stabilized. So we usually give it about 10 seconds. And then we want to, actually while we're waiting, we're going to go over here. Let me stop that again. I'm going to change this in column options to duration. And units, we're going to do minutes. And now we can hit collect, and we can save our first sample. So we'll keep our point, and now we're ready to transfer our solution back in with our algae beads, and then we'll incubate underneath our light. All right, let me switch my view. So we want our algae beads to lay in a single layer sideways if possible. So I'm going to try to shift them around a little bit in that cuvette and lay it on its side. The light that I'm using looks a little bit purple, and that's because it is an LED grow light. The advantage of this light is that we can do runs with about a 5-minute increment between samples, and we should be able to see a change in the indicator. So I'm going to set a 5-minute timer, and we're going to let that incubate. Do we have any questions?
We have one question. Pam is wondering, why did we start at 550 nanometers?
That is actually, I was going to say, if Damon wants to take this one, I can say that that is where most of the change is for the indicator.
Yeah, and that's exactly it. It's kind of the sweet spot for us being able to see it shift kind of to the yellow and to the purple. Because I know, yeah, when you look at that graph of where it's actually absorbing, right, it's not on one of those high peaks. And that's basically because that area will shift more than any other location, I think, within that spectrum.
Thank you, Damon. So to tell you a little bit more about this light, LED grow lights are becoming, I'll switch my camera view back to myself. LED grow lights are definitely becoming a lot easier to find, as well as less expensive. They also have a pretty good lifetime. When I was teaching, we went through a lot of incandescent bulbs pretty quickly because they can sometimes have a limited lifespan. They also get very, very hot and they heat up the prep. So I prefer to use LED grow lights. And the reason for that is you can actually touch them even when they've been on for quite a long time, and they don't produce very much heat. So you don't have a risk of melting any components of your lab or your prep overheating. And you have much less of a risk of course, students accidentally bumping a light bulb and maybe burning themselves. So we've got just about 3 more minutes to wait.
And also to support that, like why those LEDs are really helpful too is, I mean, we're working with algae that, you know, it usually is at the surface of a pond, so it is warm, but you can ultimately photo bleach them and kill them with too much heat. And so some of those old school, you know, incandescent bulbs that everybody's been using to do their photosynthesis labs for years, I mean, if you put that right down on the algae, it will kind of fry them. And what you'll see is they'll go gangbusters for a little bit, and then the next day they'll just not do anything, and the color of the beads will start to fade, which tells you, okay, yep, I accidentally killed all my cells. And so the investment on those LEDs is not very much from a price point of view, and the benefit is huge.
Yeah, I think I have found that these some of these brighter LED lights are enough for a whole class to share. Certainly for this lab where you really only need the cuvettes sitting side by side underneath the light, you can certainly fit a class worth of cuvettes under one single light. You don't necessarily need to have everybody in the lab to have their own LED light.
And if you're not like super pressed on time, and it's okay if that rate's a little bit slower. I mean, at some of the workshops I run, we've even just had people use their cell phone light, and what they'll do is they'll place their light sample right on top of it, just lay the cuvette right on there, and you will get change that way. It's just, you know, the rate isn't anywhere close to where those grow lights are, but you can even get away with these ones that are kind of not in the perfect spectrum. There's enough light for these beads to pick up to change. But like, once again, it's like you want to be right on top of the LED in order to make that happen.
The one light that I definitely don't recommend would be something like a compact fluorescent bulb. And in the surveys that we've done in the studies of different types of light sources, what we found is that compact fluorescents don't have enough light in the red and blue range. They are mainly in the yellow to green range, just in the, sort of right in the middle of the spectrum. So they don't really provide a whole lot of light available for plants or algae to use for photosynthesis.
Yeah, that's a good point. And some of those surprisingly also get hot. Some of those cheaper CFLs get really hot too, which you don't get a benefit any direction.
So we have another question. Why is it better to use the spectrophotometer instead of the CO2 sensor?
Well, the CO2 sensor is not made for liquids. So we do have a CO2 gas sensor, but note that it's a gas sensor. So you would not be able to use it for any type of liquid analysis. In fact, getting that sensor wet actually can burn out the lamp immediately. So you would not want to ever dip that into a liquid indicator of any kind.
I also should add to say you can do photosynthesis and cell respiration experiments with the CO2 gas sensor using plant matter. And so this is just kind of a different example of doing it by using a spectrophotometer. We actually did a previous video, Pam, about using gas sensors to do cell respiration and photosynthesis, and that should be posted on our YouTube channel and on our website.
All right, our 5-minute alarm went off, so I'm going to do a quick sample collection. So I've pipetted our indicator back out of the algae bead cuvette and into our test cuvette. And that's just 5 minutes. Keep that data point. You can see, looking at our graph with just those 2 data points, that our absorbance values are already increasing. So now we'll transfer the liquid back. All right, and we'll set those under the light for another 5 minutes. So we have had some luck. I have done this lab using the submicro cuvettes that are much smaller, and they don't allow the algae beads to fall down into the portion of the cuvette that is the narrow portion. So you can, if you want to use submicro cuvettes, you can certainly do that. You can use a fewer number of algae beads and a smaller volume of indicator. And you can even leave the algae beads in the cuvette because they won't go down into the narrow pathway. They're a little bit too large to fall down in the narrowest part of the cuvette where the light path travels. But it's a little easier, I think, to do this one.
Yeah, I was gonna say the one other caveat with that is that you'd want them to actually kind of invert it a little bit. So like, as you can see, like on my background here, you have usually kind of a diffusion problem also going where you need to get that well mixed in order to kind of get accurate readings. And so the transfer that's going on right now into the fresh cuvette actually really helps because it also helps mix everything up before you do the reading. So you get much, much better readings from it.
Thank you. I had forgotten about that. It's been a while since I did it using a submicro cuvette. And yes, I did. I had to invert it a few times gently. You want to make sure that you don't get any bubbles when you do that. But you can tap to return it. While we're waiting, I can actually show you, I've got a couple of cuvettes that I started probably about 45 minutes ago. And one was wrapped in foil so that it's in the dark. The other, I'm going to move our spectrometer out of the way. The other cuvette has been under the light. And you can see this cuvette that's been under the light is in the deeply purple range. And the cuvette that has been in the dark is pretty far over here in the yellow range. So hopefully that's visible here in our studio.
Sarah, could you stop screen sharing so they can actually see the cuvettes better?
Yeah, so this is the one that was wrapped in foil. You can see it's pretty yellow even after only about 45 minutes in total darkness. And then this one has been under that bright light for about 45 minutes. You can see how purple it is. All right, did we have any other questions?
None so far.
Okay. We'll put it back on our setup.
And one thing to know, just from looking at those two cuvettes, like once you hit those far extremes, that pH is still shifting. It's beyond where that indicator can kind of go because it's been built so tightly. So do know that if you're ever trying to like reuse a set of beads really quickly in between classes, because I've had some teachers that just want to take that purple one and then have the next class, wrap it in foil and hope it goes yellow. But if it's gone super purple, super pH high, it takes a long time to actually shift back down into that range. And that's why that washing step is so important at the start of this lab, because you want to make sure that you're kind of getting that pH range back down to the center before setting up the experiment again.
Yeah, and we're starting with our CO2 indicator as the BioRad instructions prompt you to do all of the prep steps. This has been equilibrating with atmospheric CO2 for at least a day.
And then one thing to know, if you're going to use these for kind of multiple classes, the beads survive well, just kind of out at room temp. What I would do is if I'm re-racking them, like say into that box. So a lot of times I'll just take those cuvettes when the students finish. It's still the pH indicator in there. The beads survive perfectly well in there. I'll just put them back in that foam box and then take the lid off and then just put that in a room that gets a little of ambient light, but not direct light on them. And those beads will be happy for, a good chunk of time. A week or two, if you're in between classes is that big. If you're trying to store them for longer, what I would do is I would then put them in a foam box and actually put them in the fridge, and then maybe pull them out once a month and give them a day or two someplace where there's ambient light but not direct light, just to kind of recharge, kind of rebuild some of the sugars to live off of while they kind of go into fridge sleep mode again.
All right, so we're at our 10-minute mark. Let's see, air bubble.
So Louis asked if there's already a data set out there for people to teach with. We don't have one at BioRad to share out. I'm not sure if Vernier, if you guys have any that you've collected off your SpectroVis.
I don't think we have any that we've sent out yet.
We certainly could make them available in this folder.
Especially if we save the data from today.
That's exactly what I was thinking. Yeah, we can actually make our, after our webinar ends today, I can actually do the other run going in the opposite direction. And we can have the cellular respiration data available as well as the photosynthesis data.
And since so many of you guys looks like you're still doing remote teaching, and it's kind of up in the air what spring will look like, there are ways that you can kind of do this as take-home lab stations for students. And if you need more help with that, you can email me and I can give you some of the directions that we've figured out how to kind of do this, where you can take one of these kits and then stretch them to get more student stations for kind of home use going with it, since things are still up in the air.
All right, I'm going to share my screen again so that you can see our latest data point at 10 minutes. You can see that we have a nice increase in absorbance at 550 nanometers. So this procedure is really quite easy. And the advantage of the LED lamp that I've found is that the time of incubation is pretty short. So you can do your 30-minute, you can get essentially 6 samples in 30 minutes, which is quite nice for making your graph. And then if you want to do the cellular respiration run, it's only another 30 minutes. You can certainly, if you have a short class period, you could certainly have half of your class do cellular respiration and the other class do photosynthesis and share data. There's different ways to make this short, but our goal was to use the GoDirect SpectroVis Plus and the LED lamp to shorten the time that it may take to be able to fit this into a lab period. I want to say I tested this with a halogen light as well, and it was a little too hot. The halogen lights are very nice, but you would want to use something like a heat sink if you're going to use a halogen bulb. And definitely move the prep farther away from it to keep it from overheating. So Colleen, you had passed along a question about using the CO2 sensor. We do have a lab actually that if you are looking to use aquatic plants, you can use the dissolved oxygen sensor. We have an optical dissolved oxygen sensor that's quite nice that you can use with either algae or an aquatic plant such as Java moss. And you can look at respiration and photosynthesis as well in an aquatic environment.
What I really like about using the Vernier system with the algae beads is that, especially for students that are just kind of starting down this road, it's nice because they've got a system they can visually see what's going on, and then they can easily get that quantitative value, right? So it kind of scaffolds that. And how is the spectrophotometer working? What is it producing for me? Which then makes it a lot easier, I feel like, then to move into those ones where you are using your other guys' tools where you don't have those visual pieces, but you've got just the data coming out of the devices. It's a great kind of scaffolding set for them to really be able to dig deep into these things.
Yeah, I have to say the algae beads are incredibly fun.
Yeah, the students really respond to them well just because you do have that striking color change, which is kind of fun. It's something we actually didn't anticipate, I think, when we first made this kit, that the kids would get so much into the color change. And then the fact that the beads last for a while too is kind of fun, because once you're kind of done with them, you can also send students home with them to try and put them in different places to see how they respond, over the course of time to different habitats too.
Yeah, the algae beads are so easy to handle. They're incredibly easy for students to pick up using a pipette. And you can even, if you drop one, you can even pick it up with your fingers. They're quite durable.
Yeah.
All right. right, we've hit our 15-minute mark, so I'm going to do a quick transfer here. And certainly I can see a noticeable color change in my indicator as I'm transferring it back and forth. All right, and our graph is looking wonderfully linear here. You don't always get a perfect linear graph, but this is
pretty impressive looking.
Yeah, this is looking pretty nice today. So we'll definitely share out these data with our participants today. All right, so we're back under the lamp for another 5 minutes. I'm thinking maybe we can see, I guess we have about 15 minutes left of our webinar, so we should be able to get all of our data collected.
And the beauty of having these really kind of like tight fitting graphs is that way when you move on to kind of this scaffolded inquiry, say with the students where you're changing now maybe the spectrum of light or the intensity of light, they can really like see those effects very, very clearly, which is awesome because you can really have, say, the entire class then, parse out maybe one variable instead of just, some classrooms, they kind of like open it up to everything, but you could have them do, say, light intensity by just moving the light back, a certain number of centimeters each time and really be able to see that at a really kind of fine pacing, which is, it's always the benefit of being able to use a spectrophotometer is being able to get that high granularity of what's going on.
All right, while we're waiting for another data point, I think because this graph is already so linear, I'm going to show some of the analysis features in the app that you can even use while you're still doing some data collection here. So we'll go back to our Spectral Analysis app. And you can see we haven't stopped collecting data, but we can highlight our graph here. And notice that when we highlight it, we have a number of things that show up in blue, including this little icon of a small graph in the lower left corner. That's our Graph Tools menu. And if we click on that, we can pull up a menu and see that we can use different tools. We can look at just the overall statistics that tell us things like the minimum and maximum for our data collected. We can also apply a curve fit. And let's apply a linear function to these data so far. You can see that we have an equation that pops up in this linear fit box, which gives us the slope for this line. So you can certainly have your students use the tools built into spectral analysis in order to look at the rate of change in your samples. This graph has already been auto scaled, but if you wanted to zoom to your data to get the best view, you would just click or tap on this little magnifying glass here to zoom to all data, and it will adjust it to give you the best view. Damon, I had a question for you. Can the algae beads stay indefinitely in the indicator?
Yeah, they can. Traditionally, this is where I actually store mine. And so I'll just keep them in the indicator until the next time I use them. And so I've had set go for 8 months now, just stored an indicator. The beads stay solid. And I think it's actually beneficial compared to just keeping them in water because there's more potential for them to kind of do carbon transfer being in that indicator.
Oh, that's fantastic. All right, so we're back to transfer time.
Wow, that's purple.
All right, I'm going to put a white piece of paper in here. You can see how purple the indicator has become. It started off very red from our reservoir here of indicator, and now it's really starting to turn purple, and it's only been about 20 minutes total. So we'll grab our next data point at 20 minutes. Back under the light. As Damon mentioned, this is such a great lab to demonstrate both photosynthesis and cellular respiration for students, because the time that elapses for the cellular respiration run, you do exactly the same way. You just wrap your cuvette in a small square of foil in between runs. So you can just take a small square, wrap your cuvette, and you're good to go. Can you think of any other tips or tricks that might help, Damon?
I was just going to say, since you just wrapped that in foil, from an experimental point of view, it's really important if you're going to be running those, side by side, or even asynchronously, is to put them in the exact same environment. And so I'll often have students lay those cuvettes right beside each other under the light. So even though that LED light's not, shouldn't be producing much heat, you want to have them in that exact same environment. So that really the only variable you're looking at is access to the light spectrum. And then another kind of key tip is half the reason that BioRad got those tops for the cuvettes was so that you could actually lay them on their side. So that way you get more light penetrating the beads than if the cuvette is sitting straight up underneath the light. And so that way you're usually just putting both your cuvettes, the light one, the dark one, on their sides underneath your light source in order to get this to kind of move the fastest.
That's great, thank you.
And just to kind of fill out some background on how those beads are made and what they're composed of, we're using alginate. So if you guys are not familiar with alginate, it's a polymer that polymerizes in the presence of calcium. So that storage solution that you have the beads in when you receive them from BioRad is just a calcium-rich solution, which kind of keeps those beads kind of nice and well polymerized. But I'm surprised at how well alginate sticks together. Like I said, I keep my beads after I've used them the first time just in a pH buffer, and they seem to hold up well in the pH buffer for months. So that alginate polymerization process is pretty good, pretty good.
And I think it's the same, is it the same type of alginate that's used in, you often see it on cooking shows, right? For like molecular gastronomy. It's the same kind of substance.
It's the same one, yep. So if you've ever eaten like a wedding cake with some really amazing, decorations on it, you've probably eaten this material. Or if you've eaten fake caviar, or like this new wave of gastronomy where they're just putting flavor in little bubbles like this. Yep, it's the same stuff.
I'm almost ready for our last
tech support case yet about somebody trying to eat the beads.
Oh yeah, we don't we don't want anybody to eat the beads.
So with like most manufacturing at BioRad, we try to make sure we're finding stuff that we can do these lab experiments with that are also like super, super safe, because we know students will do or dare each other to do crazy things like, you know, lick your pee glow plates and things like that in the hopes of getting glowing poop. And so we're always very keen to make sure we're trying to provide stuff the safest way as possible.
All right, we'll do our transfer again for our 25-minute mark. And we'll go back to sharing spectral analysis. All right. All right, so we're pretty deeply purple here. All right, I know that we're almost out of time, so we might want to stop, thinking I'll stop sharing. I will keep the experiment going and collect that last data point so that we can share our results out. Do we have any concluding remarks that we need to make?
We have Pam asked, does this use the free GA program or does it require GA Pro?
So this actually doesn't use GA at all. The program that we're using is free, but it's called Spectral Analysis. So if you are using the GoDirect SpectroVis Plus, you definitely want this free app from our website. And I can take you to the location to find it. All right, so on the Vernier website, first, if you need to find this lab exercise, we do have this in an article online with a short description and some links if you'd like to download more information about it. The same information is in the folder that we've shared with you today already in the chat. But if we need to find the free app, just go to Downloads. And then the app that you're looking for would be Spectral Analysis. So you can simply click on that, and that will take you to the free download page. Note that this free app is available for 5 different platforms. So you can use it with Windows, you can use it with Mac OS, you can use it with Chromebooks. So you'd want the version from the Chrome Web Store. You can also use it on iOS devices like iPads or iPhones, as well as on Android devices by getting the version that's in the Google Play Store. And those 5 different links will take you straight there. Well, with that, we are concluding our webinar. Thank you all for attending, and please don't hesitate to contact us or BioRad if you have any questions or if you need any further information.